Hi everyone. We're gonna go through some information about DNA and RNA, more just general, an overview of both of them. I'm trying to do this a little differently than I did the last remote lecture. I just feel like this might be a little bit more functional. I'm just gonna do it through a different software instead of the H5P format last time. So you'll have a paper homework assignment, which there it is. Which I'll give out after your exam. But so this is just mostly review. It's going to cover structure and function of the nucleic acids DNA and RNA, as well as covering how we have used microbiology to know a lot about what we know um, about the structure and function of DNA and RNA. So there's some experiments that used bacteria to give us a lot of the information we use today. So we're going to go through those as well. All right. So we're going to talk about these experiments that led us to our understanding of DNA and RNA, and we'll cover just mostly general structures of DNA and RNA and some differences between prokaryotes and eukaryotes. And that'll really lead us into DNA replication next lecture, which is a major target of a lot of antibiotics because there's some definite differences between prokaryotes and eukaryotes there. All right, so DNA's function is really to store the genetic information to make proteins. And proteins are such a diverse macromolecule, they give us our traits. So pigments for hair color, skin color, eye color, hormones which are chemical messengers for growth and development, enzymes are a type of protein used to make chemical reactions more efficient in our cells. We have structural proteins, muscle fibers, also cell membranes are studied with proteins to give them their structure. So lots of uses for proteins. And central dogma is just the idea that DNA makes RNA and RNA makes proteins. And a dogma is typically a term used like in religion and just for a core fundamental foundational idea. And this is the core fundamental foundational idea of biology, that DNA makes RNA and RNA makes protein. So DNA making more DNA is what we're going to focus on after today's lecture. That's just DNA replication. It's just DNA making a copy before cell division. But when we're talking about actually building a protein, we're going to talk about transcription and translation. So transcription is scribing, like taking a note of the information from DNA, the code, that was used to build a specific protein. So transcription is taking that information and just note-taking the piece of it that's needed. So it's taking DNA and making some RNA. Translation, we're then going to translate that code on a ribosome in order to direct amino acids to come in, which are the building blocks of proteins, and eventually build and fold into a functional protein. Prokaryotes and eukaryotes have a lot of the same steps for this. One of the key differences, however, is that prokaryotes, bacteria, right, don't have a nucleus. So this all happens in the cytoplasm, whereas eukaryotes, transcription will happen in the nucleus, and then the RNA will find a ribosome in the cytoplasm to do translation. So generally speaking, DNA and RNA are nucleic acids, which is one of our macromolecules, right? Proteins, lipids, carbs, and nucleic acid. Those are our main large molecules that living things contain. The NA in DNA and RNA just stands for nucleic acid, and they are both have a role in genetic information. DNA is really storing the genetic information, and RNA is then utilizing it. So DNA, the D stands for deoxyribose, which is the sugar that DNA contains. RNA stands for ribonucleic acid. Ribose is the sugar that RNA contains. DNA is the storage of all of our genetic information, so it's very big. And for eukaryotes, it's so important we keep it in a nucleus at all times. Since it has so many genes, right, so many specific segments that code for specific proteins, it tends to be large, right? Ours as humans is about 2 meters long in all of our trillions of cells. So if you were to unwind them and line them up, it can go to Pluto and back many times. It's long, but it wraps up nice and tight and condenses into chromatin and is stored in that condensed form. But DNA has the code for all the proteins, so it's the long-term storage. RNA is just the the player to then take that genetic information and build proteins. So it's the pieces that either carry the genetic information to make the proteins or is part directly of building the protein. So there's a few different types that we'll talk about of RNA. So when I say DNA makes RNA makes protein, I like to use the analogy of a really important cookbook, like grandma's famous cookbook that's been passed down, that's been passed down for generation to generation. You don't want to risk it getting ruined in the kitchen, so you're going to keep it stored safely in your office. When you want to make grandma's famous cupcakes, you're going to take a note card, like an index card, and go into the office and write down that one recipe. That index card is like RNA. And then the office is like a nucleus, right? So you're going to take that index card and go to the kitchen, that could be the ribosome, and you're going to make the cupcakes, the protein. So it's just, you're transcribing, you're taking a note of the important piece of information from all of that information, just the piece that you need right now, and then you're going to translate it into a product, which in biology would be a protein. So DNA is made out of, and I'm going to use my little laser pointer thing. DNA is made out of nucleotides. Those are the building blocks. And if it's wound up tight, it's usually stored as chromatin and then even further condensed into chromosomes before cell division. It's a little different for prokaryotes. They just have a single circular chromosome. Ours are multiple linear chromosomes. A segment of the DNA that codes for a specific protein, so a specific trait, is going to be called a gene. Sorry. All right. So DNA is storing all of the genetic information, and the genetic information really codes for proteins. And a specific area of DNA that codes for a specific protein will be a gene. We have like 30,000 genes in our genome as humans, and that ranges for different living things. So that gives us our traits and direct cell activities, and ours is stored in the nucleus, and prokaryotes just have an area called the nucleoid region. RNA is a major player in protein synthesis, so it's going to actually utilize that information that was in the DNA. There's several types. Messenger RNA, or mRNA, is going to leave, it's going to be what leaves the nucleus for eukaryotes or just travels towards the ribosome generally for prokaryotes. It is a messenger of the genetic information. So this is a copy of the DNA, the piece of the DNA, the gene, and this can leave and go find a ribosome. rRNA, or ribosomal RNA, that is what ribosomes are made out of. And tRNA is kind of almost shaped like a T. It's transfer RNA. It brings over amino acids as it's reading the mRNA. It's bringing over amino acids that will get bonded together. Amino acids are the building blocks of protein. So eventually this will be a fully formed protein. It will break off and fold into its final shape and be a usable functional protein. So to summarize, right, DNA is deoxyribose sugar, RNA is ribose sugar. That forms the backbone, the strong covalently bonded pieces of DNA or RNA. And then outside of that piece, right, there's these nitrogenous bases. And with DNA, it's where they're held together, the two strands. RNA is single-stranded, but it's built off of the DNA template, so it's going to still have complementary base pairing. DNA, we have A, T, C, and G, and A's and T's are complementary, so those bond together, and C and G's are complementary to each other. A, T, C, G, apples in the tree, cars in the garage. RNA is the same, except it has U instead of T. DNA is a twisted ladder or double helix. RNA is single and smaller. DNA has the whole code, and it's much more structurally sound, stable. Doesn't go very far from where it is, other than the nucleoid region or the nucleus. But RNA is small and just temporary and could go somewhere else. Little side step just to quickly talk about RNA viruses. All living things are made of DNA. Viruses are not specifically living things. This is one of the reasons, is that some of them contain RNA instead of DNA. So they contain inside of their capsid RNA, if we call it an RNA virus. They're going to have to utilize some enzymes because their steps are a little bit different than typically replication would use, right? Because we're starting from RNA we're copying RNA instead of DNA for an RNA virus. But they're still using our cells. They tend to, because they replicate a little bit differently, they tend to evolve a little differently as well too. They have a higher mutation rate. They are pretty diverse in terms of the type of host that they can infect. We have lots of RNA viruses. HIV is a great example. Retroviruses are just a type of RNA virus that has an enzyme that turns their own RNA back into DNA or directly into DNA, so that their host cells, so our cells, are able to replicate them faster. So just another strategy of an RNA virus to just have an enzyme called reverse transcriptase, which just reverses transcription, right? It's the opposite of transcription and goes, instead of DNA to RNA, goes RNA to DNA. So then the DNA is able to integrate into the host genome, and you're able to have lysogenic viruses that are RNA. All right, excuse me. So eukaryotic cells, like I said, have multiple linear chromosomes inside of a nucleus. Prokaryotes are single circular chromosome that are just in the cytoplasm in a nucleoid region. Now, prokaryotic cells, bacteria specifically, often have plasmids, which are just extra chromosomal DNA, typically holding something important like antibiotic resistance or things that make it more virulent, more infectious. So to summarize, right, we've got eukaryotic cells, multiple linear chromosomes inside of a nucleus, tightly wound up, no plasmids. Prokaryotic cells, single circular chromosome, just in a region called the nucleoid. Don't have to condense quite as much. They don't have proteins really that the DNA wraps around because there's a lot less of DNA. And then they often have these extra chromosomal DNA pieces called plasmids, which can get passed between different cells. Nucleotides, DNA and RNA are made out of, nucleic acids like DNA and RNA are made out of the building block, the monomer nucleotide. So their building blocks are nucleotides, and a nucleotide contains 3 parts: a phosphate group, a sugar. This is like the strongest part. This is called the phosphate sugar backbone, and then a nitrogenous base. So the phosphate sugar backbone, this is covalently bonded with a bond called a phosphodiester bond, which is really strong. Then we've got our bases, which hydrogen bond. So they're that weaker bond, so these can be unzipped for DNA replication. Let's see. RNA also has phosphate sugar base. The only difference is that instead of deoxyribose as the sugar, it has ribose as a sugar, and instead of T's, there are U's. So some people say pool, shack, barn, phosphate sugar base when you're drawing a nucleotide. And our T for DNA, but our U for RNA in terms of our complementary base pairing. And then that strong covalent bond that holds the strong backbone together of DNA or RNA is called a phosphodiester bond. And then we have base pairing between our 2 strands, A's to T's, C's to G's. You don't need to know how many hydrogen bonds. You need to know that they are hydrogen bonds, so between the 2 strands. And in RNA, instead of T, there's U, so A is going to bond with U. When we're building RNA off of a template strand of DNA, if you have DNA that's an A, it's going to code for RNA to have a U. So kind of to summarize so far, right, we've got with eukaryotes, tightly wound up DNA stored as chromosomes or chromatin, depends on what stage of the cell cycle they're in. Wrapped around proteins, a segment of that DNA that codes for a trait, codes for a protein, is a gene. And DNA and RNA are built out of the building blocks, nucleotides. And prokaryotes have a single circular chromosome as well as often plasmids, extra chromosomal DNA. Excuse me. DNA has 2 strands, and the strands build in opposite directions. So one builds in the direction called 5' to 3'. You don't need to know what that really means. It just has to do with the the ends, their chemistry of the ends of each strand. So it's beyond what you need to know. Just know that one strand we call the direction 5' to 3'. The other strand's going to build opposite, 3' to 5'. And the fact that they're anti-parallel comes into play when we talk about DNA replication, because one strand is a lot easier for the enzymes that need to build DNA to read than the other strand. How do we know all of this? Well, we're going to talk about how we know that DNA is the genetic information, the genetic material. That is thanks to some experiments we'll talk about after. But today's three-dimensional DNA double helix model is credited to James Watson and Francis Crick, who built this 3D model that won them a Nobel Peace Prize. But, you know, like many things in history, they did not credit the female behind the scenes who they took this picture from. We have this X-ray crystallography picture, which was used in order to know that we have this double helix, anti-parallel strands, nucleotides with A's, T's, C's, and G's on the ends, our nitrogenous bases. All of that is really thanks to her picture. But, you know, she did not win a Nobel Prize, and you can't get them after death. So they have the full credit, unfortunately. So our model, we call our Watson and Crick model, but we really always like to credit Rosalind Franklin in the process. What we know, right, that we have this anti-parallel 2 strands, double helix with a nice sturdy backbone that is covalently or phosphodiester bonded. And then we have our bases held together by hydrogen bonds. And, you know, gotta make a little joke about our poor Rosalind Franklin, who didn't get the credit she was due. How do we know that DNA is the genetic information? We know that thanks to some experiments that used bacteria. So we have a few here. We have Griffith's experiment first, which what he did was he noticed that some sort of genetic information, he didn't really use the term, he's a transforming principle, was being passed between bacteria. So to walk you through this experiment, we have 2 strains of Streptococcus pneumoniae, 2 strains of Strep pneumoniae. One is virulent and one is not. Our strain is called R strain because it has a rough outer coating, but it's not the virulent one. It is one that will not kill the mouse. The smooth, the S strain, is virulent and will kill the mouse. So under normal circumstances, we have the R strain of Strep pneumoniae not hurting the mouse, the S strain killing the mouse, right? Then we took the virulent strain, the S strain, and heat killed it. So now it's non-functional. However, when combined with the R strain, we know that the R strain is able to take up a plasmid that was from the heat-killed S strain. So the S strain broke down and R strain swallowed up basically the plasmids. So now the R strain is virulent because it took that genetic information from the S strain. Griffith didn't know all that, but he did notice that after heat killing the S strain and then putting it with the R strain, that now that R strain became virulent. So he knew that something was passed, but he didn't know what was passed. So our S strain was our virulent one, our R strain was not virulent, but when he killed the S strain, but then put it in the same area as the R strain took up that genetic information that was in its environment via a plasmid. Avery, McLeod, and McCarty took this a step further. So they took Griffith's experiment and just built upon it a little bit. And we're trying now, so now we want to find out, what is the transforming principle? Is it a protein? Is it RNA? Or is it DNA? So what they did was they used enzymes to isolate in our heat-killed S cells. They were able to isolate DNA, isolate RNA, and isolate protein. And when DNA was removed, when that was isolated and removed, so no DNA, it didn't work. So without DNA, we were not seeing the R cells becoming virulent, because we know that the DNA was the transforming principle, but that's how they discovered that. One more experiment just to really basically confirm that DNA was genetic information. We have Hershey and Chase doing some experiments with E. coli and a lytic bacteriophage. Now that bacteriophage, we were able to label DNA with radioactive phosphorus and label proteins with radioactive sulfur. The reason we use sulfur and phosphorus is because if you remember from biology, phosphorus is found in DNA, sulfur is found in proteins. And then when we took these bacteriophages, I'll use my pointer here. When we took these bacteriophages and exposed them to E. coli, right, the one that had the marked DNA, we were able to see the marked DNA in the next generation of phages that had been exposed to, that had been, replicated via the host cell. What we didn't see, though, was proteins showing up in the next generation of phages, which makes sense, right? Because what is being inserted from the phage into the E. coli is DNA. We know that now. And that was relevant because we know now that we're able to pass genetic information for traits to show up again in other generations. So just confirm that DNA is the genetic material. We will do this at the beginning of next lecture to practice. We'll do a review of all of this next time. But really the big takeaway from this lecture is that DNA makes RNA makes protein, and that replication is just DNA making more DNA. Transcription is DNA being copied into RNA so it can then find a ribosome and be translated into a protein. And genotype is the genetic code, right? Phenotype is the physical trait that you see. So it really makes that all connected. And here's my little analogy again with the cookbook, right? And our RNA being our note card, right? And then eventually our product being made at the end.